Welcome back, everyone, to another Explainer. Today, we're diving into something absolutely fascinating. We're zooming way in to explore the incredible microscopic world of precision microbotany. This is where scientists are literally engineering our global food security at the cellular level, and they're doing it using methods that are completely invisible to the naked eye.
So, what exactly is precision microbotany? At its core, it's the advanced application of highly specific structural methodologies to cultivate microscopic plant tissues. We're talking about biological interventions so incredibly tiny that they fundamentally redefine how we preserve, protect, and grow plant life to solve some pretty major challenges in agricultural propagation. Okay, let's dive right into this. What you're looking at here might just seem like simple, everyday laboratory work, but it represents the exact intersection of biology and engineering. It functionally holds the secrets to saving our global crop supplies from total devastation. Here's our roadmap for today. We'll cover 1. The microbotany revolution. 2. Meristem culture scale. 3. Meristem applications. 4. The embryo culture process. 5. Embryo applications. And finally, 6. Choosing the right tool.
Section 1. The microbotany revolution. The foundational divide.
When we're talking about cellular-level agricultural engineering, we're really looking at two main pillars. Pillar 1 is Meristem culture, where the primary focus is long-term viability and pathogen eradication. Pillar 2 is embryo culture, where we shift our focus entirely over to genetic rescue and hybridization.
Section 2. Meristem culture scale. The physical challenge.
0.1. I want you to just hold that number in your head for a second. We're talking about a measurement of just 0.1 millimeters, scaling all the way down from a huge field-grown plant to a microscopic fragment of this size is just a mind-blowing biological leap.
And this brilliantly illustrates the challenge. We start with a standard plant. From there, botanists extract a shoot apex that's about 5 to 10 millimeters large. But we're not done yet. From that small apex, we actually have to isolate an even tinier apical meristem measuring just 0.1 to 0.5 millimeters. That extreme, intense precision is the defining physical challenge of this entire technique.
So how on earth do we get from that microscopic speck back to a full, healthy plant? Well, it's a very precise 4-step laboratory journey. Step 1 is excision via scalpel, isolating that tiny shoot tip. Step 2 is surface sterilization. This is a chemical bath using sodium hypochlorite, followed by distilled water, just to make absolutely sure it's completely clean. Step 3 is incubation, where that 0.1 millimeter speck is cultivated in an MS nutrient medium for 16 hours under fluorescent light. And finally, step 4 is acclimatization, where adventitious roots develop right there in the tube, and the young plant is safely transferred out into a standard pot.
Section 3. Meristem applications. Solving macro crises.
Now, why does this matter? Well, the applications for global agriculture are absolutely massive. First off, pathogen eradication. By isolating just that tiny, super fast-growing meristem tip, we can effectively leave the plant viruses behind. This allows for the mass production of totally virus-free plant stocks. Second, it offers incredibly space-efficient germplasm conservation. Think about it. Instead of keeping whole fields of trees or giant expensive cold storage warehouses of tubers just to preserve a species, we can safely store thousands of genetic lines in a handful of tiny test tubes. And third, it enables the global clonal exchange of crops that literally don't even naturally produce seeds.
But that brings up a really pivotal question, doesn't it? We know we can expertly clone and clean a virus-free plant using meristem culture. But what if we want to create something brand new? How do we go about creating an entirely new hybrid?
Section 4. The embryo culture process. Cultivating the seed. This is where things get really wild. Botanists take developing seeds and meticulously, aseptically excise the young, immature embryos right out of them. By moving them to in vitro cultivation, we forcefully trigger seedling development. And there's a truly brilliant workaround here. If the standard laboratory nutrient media isn't enough to support the newly excised embryo, botanists will actually graft it onto cultured endosperm derived from the exact same species. It perfectly mimics the natural developmental support that the embryo expects.
But, as you can imagine, keeping these incredibly fragile, immature embryos alive introduces a massive biological hurdle, specifically involving osmotic pressure. There's a strict inverse relationship at play here that has to be managed.
As the X-sized embryo matures and grows, the osmotic concentration of its nutrient medium must systematically decrease.
Now, what's really interesting about this is that steep drop you see on the green line. Young, newly X-sized embryos are so delicate that they require an exceptionally high osmotic concentration in their medium just to survive and stabilize. But here's the catch. If you keep the concentration that high, development stalls out entirely. So botanists have to dynamically and systematically reduce the osmotic concentration over time as the embryo matures into a seedling.
Looking at the taxonomic origins and embryogenesis pathways, you really see how versatile this technique is. Embryos can be derived from zygotic sources, meaning from standard fertilization, or non-zygotic, apomictic sources, like somatic body cells or unfertilized eggs. Once sourced, the process kicks off with sterilization and callus induction, turning plant tissue into embryogenic cell clumps. From there, it takes one of two paths, either direct rapid shoot regeneration, or somatic differentiation, where it passes through beautifully distinct morphological phases, the globular stage, the heart stage, and the torpedo stage, right before final seedling culture.
Section 5. Embryo Applications. Biotech Interventions. This, right here, is perhaps the ultimate hero application, the genetic rescue bridge. In nature, when you try to cross highly incompatible or distant species, let's say wheat and rye, the standard nature pathway usually results in a dead end. The embryo simply aborts before it can even mature. But with our biotech intervention pathway, we forcefully extract that immature hybrid before the abortion can occur. We're literally bridging the gap, synthesizing a viable hybrid from an otherwise totally impossible genetic cross.
And heroic interventions definitely don't stop there. Embryo culture is also a total master of cycle acceleration. It completely bypasses the endogenous inhibitors that naturally delay germination, which is an absolute game changer for accelerating the breeding cycles of crops with stubborn seed dormancy, like bananas. On top of that, it allows for haploid and monoploid recovery. This is absolutely essential for isolating unique genetic lines to pull off massive intergeneric crosses, like, say, successfully crossing wheat with corn.
Section 6. Choosing the right tool. Diagnostic synthesis. When you're facing a massive biological agricultural problem, it really all boils down to your overarching goal. If your mission is to save an established plant line from disease and ensure its clonal viability, your go-to tool is Meristem culture. But if your mission is to break seed dormancy or rescue impossible, entirely new distant genetics, you've got to use embryo culture.
So the crucial point to take away is this diagnostic matrix. Just look at how perfectly they contrast. Meristem culture relies on a vegetative shoot apex just a fraction of a millimeter wide, which brings extreme precision challenges into the laboratory. Its hero application is producing those perfect, virus-free clones. Embryo culture, on the flip side, relies on reproductive immature seeds and cells. Its key lab challenge is that dynamic osmotic regulation we discussed earlier, and its hero application is synthesizing those distant, impossible hybrids.
Which leaves us with a truly vital question to ponder as we wrap up today's explainer. If these microscopic interventions can successfully eradicate devastating plant viruses and rescue impossible crop hybrids today, how will these tiny techniques continue to secure the macro-level global food supply of tomorrow? The physical scale we are working at might be microscopic, but the stakes for our world, they couldn't be higher. Thank you so much for joining me, and I hope you leave today absolutely fascinated by the invisible world powering our agriculture.